And the next speaker is Professor David Brainard, coming from the University of Pennsylvania, who is a worldwide leader of the color vision scientist and then also a psychophysics, particularly known as the co-founder of a very famous MATLAB package called Psych-Toolbox and so on. And then here, today, he's going to give a talk about the measurement and the modeling of the use of color in the material in naturalistic tasks. Please go ahead.
Thank you. Thank you, and thanks to the organizers for inviting me, and really been enjoying the beautiful city of Kyoto over the last few days. The work I'm going to talk about is done in collaboration with Anna Radnish and Nicholas Gutierrez, who are shown here.
Microphone, please.
You can't use it on? Is that better? That's better? No. That's fine. There we go. How about now? Okay. Key thing, turn it on. So I want to start with the observation that we use color and material to accomplish various natural tasks. And if we consider which tomato did we want to pick, one feature we might use is color to decide that one is likely more ripe than that. But even within the tomatoes that are roughly green, you can see apparent differences in the material, which may also be a cue to which tomato would be more delicious to pick and eat. And so the point is that both of these features, both of these percepts, help guide action, guide which one we choose. And what we're interested in this work is to develop experiments that probe the perceptual representations of both material and of color in the context of natural tasks. We want to push our measurement procedures into not just rating or matching, but more naturalistic behaviors that we do based on these percepts. And one of the reasons we're interested in both color and material is because both of these percepts are subject to some ambiguity in the world, some difficulty in having perceptions that completely map on to the physical properties. And so in color, I think most people are familiar with the problem of color constancy, where the same object with the same reflectance will change the light getting to your eye under different illuminations. This is a set of photos taken by Anya Horbert at the University of Newcastle of this structure, Uluru, out in the outback of Australia, which people go to because it's famous for actually changing its color appearance across the day, essentially defeating color constancy, because for whatever reason, this particular setting does not allow the visual system to compensate for the ambiguity caused by changes in the spectrum of the illuminant. At the same time, as we're also familiar with, and it was pointed out in some of the previous talks, image is also ambiguous about geometric reflectance. This is a nice example from Marlowe and Anderson, where you see a pretty clear apparent difference in the material of these two sides. But in fact, the pattern of light-dark vertically down the center here is identical in these two images. And if you don't have shape information, you can't disambiguate it, having to do with different percepts and different interactions between light and reflectance, depending on the perceived shape. So you have both of these percepts, although useful, are ambiguous. And so we've been interested in trying to understand how they're used jointly as a kind of prerequisite for understanding how they trade off and how the different ambiguities go. So what I want to say is that both these are perceptual correlates of object surface reflectance, but the ambiguities are complementary since one is spectral and one is geometric. The second is we're interested, as I said before, in how observers combine these two sources of information in object selection. And we want to quantify, therefore, color and material perception and their combination using a selection-based task, which is really the focus of the first part of my talk here. And I do want to point out that this work I'll talk about today builds on earlier work we just stayed in the domain of color and used selection actually in fairly naturalistic tasks. Here one where subjects had to pick a block from here, reproduce this model under a change of illumination. That work was published some years ago, and now we're trying to apply the same ideas to these issues of combination. So I'd like to tell you a little bit about the task we had subjects do, and I'll take you through the data and then a model that we use to interpret it, and maybe we'll get to some of the challenges in the discussion of that kind of model. But on each trial of the experiment, the subjects would see 3 rendered images of 3 little blobby objects, and in the center actually was always the same, and we called that the target. And on either side were 2 competitors. The subjects didn't see any of these words, I should say, on the task, just the images. And So they each differed to left and right, or they could differ from left to right. And the subject's task was a very simple one, but it was a selection task, which is to select which of the two competitors was most similar to the target. And so it's simplified from my illustrative natural task or naturalistic task. First of all, there was no need to remember the target. We showed it to them at every trial. We're using very simplified scenes, but it does preserve selection as the core thing that subjects are doing, and it's that aspect of it that I really want to focus on and how we make inferences from that. To help understand our design, I just want to now define some terminology. We had the target, and for most of the data I'll be showing you, although we had other stimulus trials in there as well, we defined one of the competitors, the color match. The color match had the same diffuse spectral reflectance as the target, so in some sense the same color, although you could perceive it differently, but it varied in its material, or in this case its glossiness, the specular highlights in it. Here much more matte than the target. And the material match was the same material rendered with the same geometric reflectance properties, but it differed in the surface spectral reflectance, so it differed in color. So we have color match and material match. And on each trial of the experiment, where the color match was and where the material match was randomized left-right, and the degree of difference was varied in a systematic way. So this slide illustrates the basic full stimulus set. And as I say in these experiments, we just stuck with one target. And we had 7 color matches, and we denote them by a kind of nominal change in material, which is a change in the specular highlight. And we go from matte to fairly glossy. There was an M0, which is the same material, actually the same stimulus as the target, and then we could step in the glossy direction or in the matte direction. We had 7 levels, of which I'm showing 4 of them here. Then, in addition, we had 7 material matches, so they had the same geometric reflectance properties, but as you can see, we got greener in one direction and bluer in the other direction by varying the matte surface spectral reflectance of the rendered stimuli. And so they vary in color. And again, there was one that was the same as the target and stepping green and blue from there. So we had this set of stimuli, and this would be presented with one of these against one of those on each trial, and the subject would say which one was more similar. So let's kind of try to go through the data representation, and I'll try to do this in a way that's fairly intuitive. And so I'm going to show over here, although I have them vertically instead of horizontally, 3 stimuli that might appear on some trials. So we have the target, and in this case, the color match is the same material. So the color match is just the same as the target. These are the same stimulus as each other. And then we've got the material matches, which vary in their color across the set of trials, and that's illustrated here on the x-axis. So C+3 is a very different color than the target. And if you were asked which of these 2 is more similar to this, it's pretty clear, since the differences are quite resolvable, that the subject is going to pick this one. And we'd expect or could plot a point at the C+3 where the subject, and this is actual data from an actual subject, always picked this color match as more similar to the target than this material match. So we've got the percent of color matched children here, the fraction, 100% at C+3. Now, if I make the material difference, or the color difference, of the material match a little smaller, this becomes more similar, but it's still different enough that the subject always picks this one as most similar. But as I keep going, now I move to a small color difference here, and sometimes the subject now picks this one as more similar because there's noise in perception and noise in the responses in the perceptual representation, and so this starts to drop down. If I go right to this case, all 3 stimuli are the same, and you have to be a chance. Actually, we don't even measure that point. We just put it there since these are just the same as each other. And then going out the other way for this, we have, in this particular case, the same pattern going the other way, although this doesn't drop down. So this curve is basically showing you the color discrimination, since here the material match is the same, and the color match has the same material. It's just a color discrimination curve. So nothing that spectacular going on with these data, but they give us a baseline for thinking about what happens next. So now let's consider the case where the color match is very different in material. So this is matte, it's got the same color as the target, and if we consider this point here, the material match is the same as the target. So you're never going to pick that one as more similar to that, and again we get this data point down here as the subject never picking this one over that one. But as we march now, increasing the color difference, now they're both differing from the target, and we're beginning to understand or see something about the way this trades off. How much color difference do we need to make up for this material difference? And this curve is characterizing that trade-off in the subject selection in this direction. And again, if we go in the other direction, we'll get a similar curve. So these two curves, or this curve now, begins to tell us something about the trade-off between color and material that subjects make when they choose the most similar to the target. We can then fill in several more cases. So I showed you the black data, and I showed you the red data. Now we reduce the material step of the color match, and the data systematically move from the red curve to the black curve. The curves now here are the fit of a model I'll tell you about a little later, just to make clearer the relationships between the point. And this is a fairly complete data set, although we also have the material steps going in the other direction. And so now we have essentially the full data from one subject. And you can see it's very regular. When you go either to the left or right, we get very similar patterns of the dashed green and the dashed and open lines. So that's fairly effective. And then in addition, we can see there's a regular progression from the large material differences to the small material differences. So that's the data. And what we'd like to do now is understand this data a little more quantitatively in terms of what it tells us about the trade-off between color and material in these selections. So just to summarize, the selection data depends on how the observer weighs color and material, but it's also going to depend on some other things, like the size of these stimulus differences. We don't have adequate stimulus metrics to really predict those differences either in color or material in a way that's precise enough to allow us to count on having equal steps, and particularly to have equal steps between the color and the material dimensions. And so we needed to develop a model that's going to recover both the perceptual stimulus steps corresponding to the stimulus set, as well as a parameter describing how observers weight the two variables, one against the other. And that's going to be the next bit here. And this model is an elaboration of something that's called maximum likelihood difference scaling. It was developed by Larry Maloney and colleagues. And you can think of it as combining ideas from really two fundamental kinds of perceptual modeling. One is multidimensional scaling, and the other is the theory of signal detection. So I'll take you now through the modeling ideas and how we fit that model to these data, actually how we got these curves here, and then how those fits allow us to answer the question we set out to answer at the begin with. So in our model, we say that each stimulus is going to be represented by a point in a perceptual space. And so we could imagine a color axis and a material axis, and we had these 40 odds, these sets of stimuli, 7 by 7 stimuli. And so there'll be a color axis going this way, and this would material axis, and these are meant to be perceptual representations. So we don't actually know that they are equally spaced, and we know that they're noisy. So the representations are going to have a little cloud around them to denote, and this is sort of the idea from the theory of signal detection, that there is noise in the perceptual representation, that the draw on any given trial may not right be at the center of this, but could be spread out according to some distribution near there. And in addition, we don't know that our spacing along the color axis and our spacing along the material axis are the same, so these one axis could be stretched perceptually relative to the other. And finally, we don't know that the spacing is uniform along the two axes, so there can be some variability. These are wherever they are, and those are wherever they are. But we're going to assume that that representation is underlying observers' choices. It's just that we don't know for our stimuli where to plot each stimulus. And given that there is a representation like that, then we need a theory that's going to link the representation back to perceptual performance. And so the idea is that on each trial, the actual stimulus representation is a noisy draw from the distribution describing the stimulus. So the target might not be right there, it might be shifted over here. The color match might be shifted this way, the material match that way. And so the observer is faced with an internal representation on each trial for these three stimuli here that might be on that trial, something like that. And our linking hypothesis between this construct and performance is that the subject chooses the stimulus as most similar to the target that's closer in the actual representation, perceptual representation for that trial. So there's a distance computed between the target and the color match, and the target and the material match. And that distance, if we had no differential weighting, would just be the standard Euclidean distance, and calculated like this. And then the last part of our theory is subjects may weight color and material differentially in their decisions. And so we need a weight factor, a parameter that describes the relative weighting of the color dimension color difference, and then 1 minus that is the weighting of the material difference. So the parameter W is the color material weight. It's actually what we set out to understand, now expressed as a parameter of the model in terms of how subjects are trading off these two aspects of perception when they make these selections. So we can actually take that model, since it makes a prediction for what happens in every trial, and we can literally fit it to the data by choosing the positions of each stimulus along the two axes, choosing the weight, and then from that, by simulation, we can run out and make a prediction of how, when we pair any one of these stimuli against any one of those, what the fraction of the color match chosen ought to be. And these curves, to the same data set I took you through before, are in fact the fit of this solution directly to these data. And so we see a series of positions here, and they're not quite equally spaced. They're roughly the same extent expressed in units of the noise along the two axes, roughly equally spaced, although not quite. And this subject is trading off color and material nearly equally with a color material weight of very close to 1/2, putting equal weight on those two axes. Now you could wonder, and we certainly spent a long time wondering, how good a fit is this? And so one thing you can do is for each trial or each trial type, you have a measured proportion of color matches selected, and then you have predicted by the model. And you see the model is doing a very good job here of actually accounting for the full data set, of which I've shown you just a slice in my expository description of the data. So then we can do this with other subjects and ask what happens. And so here's another subject, and we see a couple things. First of all, now for this subject, the same set of stimuli are spaced more broadly along the material axis than the color axis. So the perceptual representation of this subject differs. But in addition, these are independent and identifiable or more or less identifiable parameters of the model. This subject places more weight on color than on material, has a higher color material weight than the other. And you can see the pattern of the data is a little steeper here. That's the sort of explicit data-based expression of these two facts. But the model allows us to tease them apart and solve for them simultaneously. So we repeated this procedure for a number of naive subjects. And a kind of striking feature of the results is a difference in terms of how they weight these two aspects of perception in this very simple selection class, ranging from the subject I just showed you at 0.75, a subject down here, the first one at about 0.5, and down to one who weights almost the inverse of this two extreme. These error bars are from a bootstrapping analysis of the precision with which we could recover these parameters. And although there is some, it's hard to lock them down completely in the presence of uncertainty about the perceptual representation, it's fairly clear these confidence intervals are small enough that this subject, for example, is different from that subject. I should say we subsequently redid this entire experiment with more stimuli and kind of fancier adaptive psychophysical procedure, and saw the same kind of pattern of variability in this parameter, which was published about a year after this study here, but I won't take you through that today. So from the experimental part of this work, I'd say that the conclusions I'd like you to draw are, first of all, that we have actually developed a procedure that allows us, using selection, to measure color material weighting for object identification. And we were, as I said at the outset, particularly interested in doing this from selection because we wanted to study both superthreshold stimuli and also to study the task that we think is the one that observers primarily use these attributes for in much of daily life, which is to actually make judgments about what's yummier or what's preferable across various things. And we heard yesterday a number of the ways in which in the consumer literature these kinds of attributes guide people's choices. And so now we have got an experimental method that connects that task, that kind of core task, to the underlying perceptual representations and trade-offs. That's based on a model, which is a 2D generalization of maximum likelihood difference scaling, and that recovers the stimulus differences in the subjective perceptual space, along with the color material weight. The weights vary across observers, but we can now study, at least in principle, study how these are affected by other stimulus things. So for example, we might imagine introducing variations in the spectra of the illumination, but not in the geometric properties, and asking whether that shifted a subject's color material weight as there was more ambiguity in the color representation than perhaps the material representation, or vice versa. Those were the sorts of experiments that we'd like to drive toward on this program, to really understand how the constancies or lack of constancies in these two complementary object properties might interact. And then I would say that finally, and this comes back a little bit to the question that was asked in the previous talk, this work provides us with a functional or computational characterization of how the stimuli are represented and how they're traded off in this particular judgment, but it doesn't tell us by itself anything about the underlying mechanisms that are mediating that visual representation or those trade-offs. And that's something I think all of us, in addition to wanting the computational description, would like to have. So in the second part of my talk, I'd like to turn a little bit to some computational work we've been doing that takes what I'd call a baby step in that direction. It's not based on measuring the end product, but rather based on trying to start at the beginning of vision and carefully model what happens to the stimulus as it passes through the early stages of vision, and ask how do those early representations govern or not govern, or reflect or not reflect the kind of performance we see in the human observers. And so I'll tell you a little bit about that project. We refer to this project as, or the software that's underlying it, as the Image System Engineering Tools for Biology, or ISET Bio. And this is a work I've been doing primarily with Brian Wandel. Actually, I trained with Brian Wandel a frighteningly long time ago, and for many years, although we remained good friends and talked to each other about science, we didn't collaborate together. But more recently, we've come back and sort of teamed up on this software with a series of postdoc students. Nicholas Kateris has been really key on this as a PhD level senior scientist in my lab, and some collaborators and other students who've helped us move along. And the goal of this software is to encapsulate, really in an image computable way, the representations and what we've learned over actually hundreds of years now of vision science about how light reaches the retina and how it's transduced by the cones and so forth. To say that more explicitly, it's a set of computational tools designed to model how light from the scene is encoded and processed in the end by visual circuitry. And we're starting at the beginning and working our way in. And as you'll see, we haven't gotten even past the first synapse yet, but there's still a lot of richness there to think about. And a particular goal of this project, which I'd like to pitch to you, particularly if you're interested in these matters, is there's a lot of knowledge in what are the physiological optics, what's the scene flow, what are the photoreceptors, how are they packed, how does the eye jitter, and it's hard to go to the individual papers, and if you want to use it, do that all yourself. And so the idea that there might be a reusable software package that would encapsulate that knowledge seemed to us a very good idea. So ISET Bio is open source. We happen to have written it in MATLAB. The distribution, which is on GitHub, you can get there via this link, contains tutorial code, and it contains various validation and unit tests and regression tests that we have done our best at least to make sure that the bugs that remain are small. I think you never really get rid of all the bugs, but we beat down as many as we can. And it really helps clarify, we think, the impact of the different elements of early vision on visual performance. And what I'll tell you about now is what those elements are that we've modeled, and what they say or don't say about the task I've just described to you. So ISET Bio has a series of components, and in early vision, it's fairly feed-forward, so we can be reasonably clear about that. And since we're talking today about visual psychophysics, one of the things I'll emphasize is we have display models that characterize the properties of typical displays, in a particular spectra of the channel primaries and the gamma function of displays. And we know in this experiment what display we used and what its properties were. And so we can specify the input to the visual system for our experiment quite closely as input to ISET Bio on a display model. Actually, I should say, we've worked hard in this software to always stay in physical units. So we specify spectral radiance when we talk about display primaries and stimuli. We specify visual angle. We specify retinal radiance in, again, physical units and so forth, which we found very helpful in clarifying just to think about the magnitude of the signals and those kinds of things. Now, the image from the stimulus is then imaged on the retina. If it's a flat display and we're talking about sort of the central fovea, we can model that as blurring by point spread function, and there's been great progress over the last 20 years in measuring and characterizing the point spread function of the human eye using wavefront optics. And so we've taken Tybos's data set, which characterizes the eyes of 200 individuals as a polychromatic point spread function. And one of the things that's striking when you do that is how much blurrier the chromatic aberration at the short wavelengths, 410 nanometers, compared to an accommodated wavelength of about 550, is spectacularly enormous. And it's fairly large at longer wavelengths as well. And so by starting in a spectral representation, we can capture the differential blur in the formation of the retinal image. I should say we also have, and for cases where we're studying 3D scenes is quite useful, a ray tracing solution through a model eye based on PBRT, which you heard about a little bit yesterday, that accounts for depth of field and transverse chromatic aberration, et cetera, and that's the work of Tricia Lian. It was just published in the Journal of Vision. Today we'll be using the point spread function model. So then the next thing that we characterize is actually the locations of the cone mosaics. There are 3 classes of cones with their distinct spectral sensitivities. The effective sensitivities of the S cones are much smaller, largely due to inert pigments in the lens and in front of the fovea. The packing geometry of the cones is quite interesting. There's an S cone free area in the center. LM cones are fairly random. S cone packing is a little more regular. You can't quite see them there. They're very small dots. And we actually have an algorithm that generates model mosaics that model the fall off in cone density with eccentricity. And we try to get this stuff as right as we can. And so we have the cone mosaic. And from that, we can calculate the isomerization rate of the photopigment in the individual cones in the mosaic. Other components that are in the software that I'm not using today are a model due to Engbert and Ralph of fixational eye movements, particularly drift eye movements, a model of the photocurrent transduction, which has a nonlinearity and a temporal filter. And we're beginning to work on now the retinal ganglion cells, but I wouldn't say that that's very mature yet. Up to here, the software is quite mature. We have a bioarchive paper describing these components and their application to understanding human contrast sensitivity. So for this project then, we started with our stimuli, each stimulus, and we passed it through the model of the optics to get a retinal image formation. This yellowing here is due to the absorption of light by the lens of the eye, which filters out short wavelength light. It's actually quite, if you just render it like this, quite an enormous effect. Obviously not perceptually salient because the brain has grown up dealing with that. And, but there is a real transformation in the spectra as you arrive at the retina. And then to the isomerization rate, or mean number of isomerizations in each cone. It's a little hard to see here, but each cone is a little dot. They're all gray because cones are univariate. They are not themselves colored in any way. They just produce signals. And the little dark ones are the S cones whose isomerizations are lower. You can't really resolve that here. And for this particular experiment, we modeled 15 by 15 degrees of visual angle, which was the size of the stimuli. And so there are 277,059 cones in that model mosaic. And so each stimulus, basically, as is passed through early vision, is now represented by a very high dimensional vector. And as I say, we didn't include eye movements or photocurrent in this modeling. And what we can then do is say, well, if that's the visual representation of each stimulus, and we're trying to model this task at that very early stage of visual processing, then we can simply ask, well, we can use the same principle we did in our perceptual model, but now the mechanistic model is saying, this is the representation, I've computed it, what's the distance in that space? So a space where each axis is the response of a cone. And we know that there's Poisson noise in the cone photoisomerizations. That noise is very small at photopic light levels relative to the variability of performance. And so we do have a parameter in the model, which is isotropic Gaussian noise, used to bring the variability in the model responses based on the same distance principle as we used in the perceptual model into alignment, broadly speaking, with human performance. And for this particular simulation, we weighted, we didn't really have color and material. We just weighted each dimension equally in the computation of distance. So that allows us then to simulate out data on the assumption that the mechanism mediating this, and I'll say it's not a very likely to be correct assumption, but it's the assumption that this is the representation that directly mediates performance. We can calculate what a performance would be. And then we can fit that data, so that's the simulated data in the same format I showed you before. I didn't put the point right at 50% there, but there'd be one. And you can see it's a little more asymmetric than our human observers, and it has a slightly different shape. And the solution, since we didn't build any asymmetry in, recovers a color material rate of about 0.5. And again, you can see that there's a shift, that the stimuli are not centered in the way they were for the human observer. There's a difference there and a difference there. So this model tells us what we would expect if we use the same kind of decision rule we think mediates perceptual performance, but apply it at the level of the cone isomerizations. And I would say it does not predict many features of human performance that we observed in our observers. And that shouldn't surprise us, because we do think there's retinal processing going on and things happening after the cones. For example, color opponency is not included in this model, but it's known to be fundamental to color discrimination. We haven't talked about the retinal ganglion cells. We haven't talked about the cortex. And so you might say, well, why did we bother to do this? I think the reason is that we know 100% for sure that the visual signals pass through these initial stages. So the effects of these things are obligate. You can't get around them. And we might as well understand what their implications are to define the problem that's faced by the rest of visual processing. And as this kind of modeling progresses further into the visual system and is complemented by measurements of what's actually happening in responses further along pathways, we hope to bring this progression from the outside in closer and closer to actual neural mechanisms and their description. And one of the things to say, though, is that the principle that we've built in our perceptual model and the principle that we've built in the representational model here of using the same distance ideas to predict performance put the two kinds of models into the same units. That is to say, we've taken our ISET biocalculations and produced simulated data that's in the same format and can be solved and analyzed in the same way as the psychophysical model. And that's an explicit linking hypothesis that we're making here, which if you don't like it, that's fine, but we've made it quite clear what it is and allows us to proceed in making connections between these two domains of inquiry, one representational and one derived from the psychophysics. So I will just show a little more of that sort of model solution and two subjects before, again, emphasizing some of the qualitative differences here. The perceptual representation from the cone isomerization, as I said, differs from that of the human subjects, and not surprising, but made explicit. And our longer term goal is to go down this road of pushing further in. So I showed you those conclusions before. I've graded them out. I'll just finish by making 3 points, which I've probably already made, but as they say it again at the end. We're approaching mechanism by building on fundamental measurements of how early vision works. And it's implemented as open source code. It's modular, and they're explicitly computable. And we're planning to move step by step further into the visual system, with the bipolar and ganglion cells of the inner retina being next. And although there are surely important processes that follow early vision, the information loss and the representations that it produces are the input and define the computational challenge for the rest of the nervous system. And we'd really like to understand and be able to do that for any psychophysical experiment that we're considering. And finally, ISET Bio is really intended to make these calculations easy enough so that other people who might want to use them don't have to go and do it again from scratch, but can build on this and do the same kinds of considerations if I've persuaded you that they're useful. So I will stop there, and I'd be happy to take questions.
Questions?
So I have a bunch of questions that I'll leave for offline, but two things. One is, does the distance between the choices make a difference? Because in our monkeys, we've discovered that what we think is that even that brief amount of time that it takes to make a saccade from one object to another, we see increases in the time it takes if the objects are moved further apart during choice. And we think it's because of very short-term memory effects. And so the question is, have you measured reaction times of the subjects and looked at how that varies with the difficulty of the precision with which you're asking them.
Yeah. So the answer to the actual direct answer to your question is easy. No, we haven't measured the reaction times. I think a really interesting direction for these kinds of experiments is to move to tasks that actually explicitly involve memory. Since I happen to have it here, this is sort of our vision of where we might imagine going with this kind of choice experiment as we try to put it more and more into naturalistic context. You can imagine you're playing some video game. There are friendly dragons and mean dragons that steal your gold, and they're a different color. You have to both learn that, and you encounter them under different lightings and different circumstances, but you can make the core selection now based on a memory standard. That's an idea that we think would be exciting to pursue, to kind of systematically do that. We also could obviously, in even the simple characterization, introduce a delay between the standard and the tests and do that. But what I've described is as far as we've gotten.
You're going to find interesting things when you do it. I'm sure of that.
This was quite fascinating. I was intrigued by the very large variation across subjects. So first, I assumed that within subject, people were consistent. I assumed those 5 subjects are the same age, gender, more or less, and more or less. And then, how do you think about it? In particular, connecting to the second part of your talk, I would imagine that these kind of models would not explain that kind of enormous variability.
So the subjects are all basically young adults. Some are men, some are women. The differences are quite reliable. That's something we spent a long time on, and as I said, we did an additional experiment with a different set of 9 subjects showing the same kinds of range of performance. And so now the question of what's driving that becomes quite interesting. I think that within the modeling, and again, this modeling is obviously the early visual system, the obvious differences, and I'd be shocked if they accounted for the actual subject differences, would be differences in the optics, and differences in the L to M cone ratio, which are known to be large, maybe minor differences in the spectral sensitivity. And in principle, you could characterize these things on a subject-by-subject basis, although it's quite a lot of work to do so. I suspect that these differences are due, therefore, to processes that are later on and have to do either with just experience with these stimuli or with just literal differences in the cortical representation, but I don't have anything very concrete to say about that. But I think it's an interesting question, for sure.
Short question, please.
The first thing is about the orthogonality, or you assume that the material dimension and the color dimensions are orthogonal, but it's not. Even in a for example, microprocess theory starts. Physically, it's not. They are not independent. Perceptually independent.
This is something we spent a long time fretting about, and I'll take a stance on it, but you could push me off this stance pretty easily. The problem we face, and I should say the problem we face with this kind of measurement is that as you introduce more parameters into the observer model, the amount of data you need to lock them down becomes larger and larger, and it's a losing proposition. And so you sort of have to, we found this was as far as we could go. If we either allowed, for example, the noise to be anisotropic or the dimensions to interact in the representation, then what I want to say, then changing one of those parameters really would move around values of the other. So I think when you think about the solution, you have to think about it rather explicitly in what set of assumptions did we make, and then what's revealed within those assumptions. And if the assumptions are wrong, then you're going to have to either find more ways to come at the experiment to constrain it, or you're going to have to find a different set of assumptions that are equally powerful that are right. I don't think that's, so the stance I want to take is, I don't think that's bad. I think that's the way we progress, but it's something to be aware of.
Other question is about when you directly apply the ISET bio to the real situation, but you don't know the parameter of each of individuals.
That's correct. So this is a generic model, and I would say is that the generic model, which was kind of typical optics, typical cone ratio, typical cone density, didn't look a whole lot like any of our subjects. But one could, and I'd say this is one of the powerful things about ISET bio, is we could systematically have moved those things around and asked how much did they matter? And what I would say is the tool gives us the ability to do that in a way that if I were a better person, I would have done it and checked my intuition that it wouldn't have a lot of effect.
Thanks.
Okay, it's time again. Thank you very much.